Okay, so welcome back to this next video in which we are discussing the LDL receptor. Okay, right, so I promise you we will get to LDL and the LDL receptor soon. I just want to make sure that you do know where LDL actually comes from, because we're going to see that basically it's the remnant of one of these VLDL molecules. Basically, when you take the VLDL and you remove all of the triacylglycerol molecules from the VLDL molecule, then what you get is an LDL molecule. But we'll come to that in a moment. Okay, right, so we have had the liver package up triacylglycerols into these very low density lipoproteins, which have then been secreted into the blood. And what's now going to happen is these very low density lipoproteins are going to go to the capillaries within skeletal muscle cells, well, skeletal muscle tissue, and also cardiac muscle tissue. Okay, and the endothelial cells of capillaries within skeletal and cardiac muscle tissue are going to have special apparatus on their apical surface, which is going to allow them basically to unpackage the very low density lipoproteins. So let's show this on the next page. So, if we show one of these endothelial cells of a capillary within the skeletal cardiac muscle cell here, then I can show the special apparatus that it has on its apical surface. So basically, on the apical surface, you have a protein attached to the apical membrane of the endothelial cell. Okay, so it's not a transmembrane protein, it's not integrated into the membrane. Instead, it's attached by a group known as a glycosyl phosphatidyl inositol group. Okay, so I would just like to discuss with you what a glycosyl phosphatidyl inositol group is. And luckily, because of our extensive discussion of the structure of lipid molecules, we're actually in a good starting position to understand what a glycosyl phosphatidyl inositol group is. And by the way, glycosyl phosphatidyl inositol groups are often abbreviated to GPI, and they're a common group that you can add on to proteins and which will allow them to be attached onto the outer surface of cell membranes, like indeed this protein is going to be. And we'll come back to the name of this protein once we've discussed what glycosyl phosphatidyl inositol is. So, we've got our old friend here, phosphatidyl. That means that this structure, whatever it is, must be based on the structure of a phosphatidic acid molecule. So let's start by drawing our old phosphatidic acid molecule. Okay, so remember we have our two long chain carboxylic acids, okay, which I'll denote in orange here. We then have our glycerol molecule, which I'll denote in green here. And we then have our phosphate group attached via a phosphester link to the alcohol group that comes off the third carbon of our glycerol molecule. Okay, right, so that's our phosphatidic acid molecule. What we now want to create is phosphatidyl inositol. So we're going to worry about the inositol before we worry about the glycosyl. Okay, so inositol then. Inositol is the old biochemist's name for a molecule that would now be called cyclohexane, and then 1, 2, 3, 4, 5, 6 hexol. Okay, and although, yes, that's a mouthful, it's a useful name because, again, it tells you exactly what you're dealing with here. It tells us that we're dealing with a 6-carbon cycle, basically, so a hexagon of carbon atoms, basically, where all of the bonds between the carbon atoms in that hexagon are single bonds, okay, and off every single carbon you then have a single alcohol group, which means that every single carbon also has a hydrogen coming off it. Now basically you are going to attach the alcohol group coming off one of these carbons to the alcohol group of the phosphate group of the phosphatidic acid molecule via a phosphoester link. Okay, so here is our inositol ring, which I'll just denote as this blue hexagon here. Okay, and every single carbon will have an alcohol group coming off it, and that's how we have linked this inositol ring to the phosphate group via a phosphoester link. Okay, so that molecule then is now called phosphatidyl inositol, and it's an example of a phosphoglycerolipid. Okay, now we want to turn this into glycosyl phosphatidyl inositol, and to do that what we're going to do is link off some sugar molecules onto this phosphatidyl inositol structure. So whenever you see glyco, that should instantly cry out to you sugar. Okay, so what we're going to do is link off the alcohol group coming off this carbon of the inositol ring here. We're going to link a few sugar molecules. Okay, specifically we're going to link 4 sugar molecules. 1, 2, and I'm just going to denote these sugar molecules as boxes. Okay, 3, and then finally 4 here. So the first one here is going to be a glucosamine sugar. Okay, which means basically take the alcohol group off the second carbon of the glucose ring and then replace it with an amino group. That's how you turn glucose into glucosamine. So this is glucosamine, and then the other 3 here are all the same, and they are mannose sugars. Okay, now mannose is just an optical isomer of glucose. Okay, that has a different name. So these are all mannose sugars. Right, so we have a glucosamine sugar followed by 3 mannose sugars, and then finally attached to this what you have is another phosphate group. Okay, so this is a phosphate group here, finally attached to that final mannose, and then attached to the other alcohol group of the phosphate group via a phosphoester link. You then have a structure known as an ethanol amine group. Okay, so here is the ethanol portion, which has the 2 carbons, and then the alcohol group is involved in the phosphoester link to this phosphate group, and then here is the amino group. Okay, NH2 here. Right, so this whole structure here, when you have the phosphate group attached via a phosphoester link to ethanol amine, is called phosphoethanol amine. Okay, so we could say that the glycosyl phosphatidyl inositol molecule consists of a phosphatidyl inositol molecule with these 4 sugars, glucosamine followed by 3 mannoses, then linked to a phosphoethanol amine molecule. Now, this is a beautiful lipid molecule which will quite happily sit within phospholipid bilayers of cells. Specifically, it likes to sit in the outer layer of cell membranes, so it will sit in the layer that faces out towards the external world. Now, basically what you can do is you can attach these molecules to the carboxylic acid terminus of proteins. So if I have a protein here, let's say this is the amino terminus of our protein, and this is the carboxylic acid terminus of our protein. So remember, proteins fundamentally are just polymers of amino acids. So the first amino acid has a free amino terminus because its amino group didn't need to be involved in a peptide link to the amino acid before it because there was no amino acid before it. Similarly, the final amino acid in the polypeptide has a free carboxylic acid group because its carboxylic acid group didn't need to be involved in the binding to the amino group of the next amino acid along. Okay, so what you can do is you can attach these glycosyl phosphatidyl inositol molecules onto the carboxylic acid terminus of these polypeptides, well, of certain polypeptides. Not every polypeptide gets this happening to it, but certain polypeptides have this happen to them. So here's the polypeptide here. Okay, and basically what you're going to do is you're going to form an amide link between the carboxylic acid group that's at the end of the polypeptide and the amino group of the glycosyl phosphatidyl inositol. So you will take off one of the hydrogens of that amino group, and I wish now that I had drawn this amino group like this rather than as an NH2. So there we go, I've just replaced the amino group like so. And what you'll do is you'll take off the alcohol group off the carboxylic acid molecule, you'll take the hydrogen off the amino group, you'll bind those together to make water, you'll then bind the nitrogen of the amino group to the carbon to create an amide link here. So this link here is called an amide link. Okay, right. So that's how you can attach proteins to glycosyl phosphatidyl inositol molecules, and the significance of this is that this protein here can now be attached to one of these glycosyl phosphatidyl inositol molecules that is sitting within the outer layer of lipids within the lipid bilayer of the cell membrane, and it is therefore attached to the outer side of the phospholipid bilayer basically by being attached to one of the lipid molecules which is within that outer layer of phospholipids. Okay, right. So, what is this protein then? What is the name for this protein? Well, this is why I wanted to discuss what GPI actually is before I gave you the name of this protein, because this protein is called GPI-anchored. Okay, so that's the first part of its name. And then HDL, high-density lipoprotein, binding protein. Okay, and specifically it's the GPI-anchored HDL binding protein 1. Okay, and for short, that is abbreviated to GPI for GPI-anchored, then H for HDL, then BP for binding protein, and then you put a little subscript for 1, so the GPI HBP1. Okay, right. So, as I say, these proteins are not transmembrane proteins. They are held at the outer surface of the phospholipid bilayer of cells via these GPI anchorings. So, basically what happens is these molecules dimerize. So let's say this is another one of these GPI-anchored high-density lipoprotein binding protein 1s. Okay, they dimerize together on the outer layer of these endothelial cells within cardiac muscle tissue and skeletal muscle tissue. And then this dimer of GPI HBP1 proteins then binds another dimer on top of it. Okay, so on top of this, we have a little dimer on top here, which is called the lipoprotein lipase homodimer. So in blue here, this is LPL, which is short for lipoprotein lipase. So the first L is for lipo, the P is for protein, so lipoprotein, and then the second L is for lipase. And this is specifically the lipoprotein lipase homodimer. Now, basically, to function, a lipoprotein lipase needs to dimerize like this in a homodimer. And let me explain to you why. Okay, so basically, lipoprotein lipase I don't think has been crystally structured yet. Okay, so we don't have a crystal structure of lipoprotein lipase as far as I'm aware yet, but we do have a basic model of its structure. Okay, which consists of splitting its entire structure into two separate parts. Okay, so a portion that's near the amino terminus and is therefore called the amino terminal domain, and then a portion that's near the carboxylic acid terminus and is therefore called the carboxylic acid terminus domain. Now, basically, to make a functional lipoprotein lipase enzyme, you need both this amino terminus domain here, which I'll colour in blue, and you also need this carboxylic acid terminus domain, which for a change I'll colour in yellow. Okay, which probably isn't going to show up at all in this light, but never mind. Okay, so you might think, well, surely the enzyme can function on its own, but basically this amino terminus domain and this carboxylic acid terminus domain in the same protein, they find it difficult to work with one another. Okay, they're not in the right positions relative to one another. So what happens is you get another one of these proteins and you put it basically the other way round, like so. Okay, so that, oh no, what have I done? No, that shouldn't be a carboxylic acid terminus, this should be an amino terminus. Right, so that the amino terminus of this second lipoprotein lipase is in the correct position to interact with the carboxylic acid terminus of that first lipoprotein lipase. And the carboxylic acid terminus of this second lipoprotein lipase is correctly positioned to work with the amino terminus of the first lipoprotein lipase. So this here makes a functional enzyme, and this here also makes a functional enzyme. So that's why you have to dimerize them together, even though they have the necessary portions to make a complete enzyme on their own, because on their own they just can't get into the right conformations relative to one another. Whereas when you join two of them together, that makes two functional enzymes now. Okay, right, so that's why you need to dimerize lipoprotein lipase proteins together like that. And that's what this structure sitting on top of the GPI-HBP1 homodimer here is doing, basically, why that's important. Okay, so only endothelial cells within skeletal or cardiac muscle tissue will have this assembly on their surface. Okay, now what's the significance of this? Well, basically, now comes along our very low density lipoprotein. Okay, and basically what's going to happen is the lipoprotein lipase homodimer is going to stick itself into the phospholipid monolayer of the very low density lipoprotein. So let's just remind ourselves of the structure of our very low density lipoprotein. So this outer layer, remember, is the phospholipid monolayer, and now the lipoprotein lipase has stuck itself into that. And then in the center, you then have this core, basically, this hydrophobic core, where, remember, the triacylglycerol molecules are stored. So remember, these are these molecules where you have glycerol bound to 3 long-chain carboxylic acids. And you also have, less importantly here, but you also have those cholesterol esters, okay, where you have a long-chain carboxylic acid molecule attached to a cholesterol molecule here, which I'll have in turquoise. Okay, right. So, what you're now going to do is the lipoprotein lipase, which has stuck itself into the very low density lipoprotein here, the VLDL, is going to bind to a certain apolipoprotein which is within the phospholipid monolayer of the very low density lipoprotein. And the binding of the lipoprotein lipase to this apolipoprotein is going to activate the low lipoprotein lipase. So, which apolipoprotein does the lipoprotein lipase bind to? Well, basically, it's apolipoprotein C2. So when I told you the 3 main apolipoproteins which were present within the very low density lipoprotein phospholipid monolayer, apolipoprotein C2 was one of them, okay, along with apolipoprotein B100 and apolipoprotein E. So, one apolipoprotein C2 molecule will bind to one of these lipoprotein lipase molecules, and another will bind to the other. Okay, and when the apolipoprotein C2s bind to the lipoprotein lipase homodimer, what will happen is this lipoprotein lipase homodimer will become active, and what's it going to work on? Well, it's going to work on the triacylglycerol molecules. Okay, so what does it do to these triacylglycerol molecules? Well, basically, it's firstly going to hydrolyze the ester link between the long-chain carboxylic acid and the glycerol molecule on one side. Okay, so let me show this. So basically, when lipoprotein lipase splits triacylglycerols apart, it goes for the ones on the ends, basically, and leaves the one in the middle. Okay, so here are the long-chain carboxylic acids in orange here, and here is our glycerol molecule here. Okay, so this is our triacylglycerol. What's going to happen is, firstly, lipoprotein lipase is going to break off the long-chain carboxylic acid on the side, basically. So it's going to start with the side, and it will create a diacylglycerol that looks like this. Okay, but it doesn't stop there. It can go further than that. It can cut off the other one on the other side now, okay, to leave only the one in the middle remaining. Okay, and of course you'll get a free fatty acid as your other product here, so this is a free fatty acid. Okay, but it's going to continue on, so this reaction was catalyzed by lipoprotein lipase, and this next reaction is also going to be catalyzed by lipoprotein lipase. And what you're going to end up with is a single long-chain carboxylic acid, which is esterified to the alcohol group that comes off the second carbon of the glycerol molecule, okay, like so. And this is called a 2-monoacyl glycerol. Okay, so the 2 reminds us that the single acyl group comes off the second alcohol group of the glycerol molecule. Okay, and when you perform this cleavage of the diacyl glycerol molecule, you'll also get another long-chain carboxylic acid molecule released. So you get another free fatty acid. Okay, right, so that's what the lipoprotein lipase is going to catalyze when it sticks its face into the hydrophobic core of the very low density lipoprotein. Okay, and what this is going to result in is the production of these 2 monoacyl glycerols, and also with the production of 2 free fatty acid molecules. Now, lipoprotein lipase can't go any further. It can't split the glycerol molecule and this middle long-chain carboxylic acid apart, but it doesn't matter, because what can now happen is the free fatty acids can leave the hydrophobic core of the very low density lipoprotein, and also the 2 monoacyl glycerol can also leave the core of the very low density lipoprotein. And what will happen is they'll diffuse out of the very low density lipoprotein, they'll diffuse across the endothelium, and they will then go to the tissue, which of course is the skeletal muscle tissue or the cardiac muscle tissue, and then the cardiomyocytes will use them for energy basically. So you're taking 2 monoacyl glycerols and 2 free fatty acids here. Okay, so here are the 2 free fatty acids. Here is a long-chain carboxylic acid still esterified to that second alcohol group of the glycerol molecule, and both of these are moving into the cardiac muscle tissue or the skeletal muscle tissue, and then what will happen is they will be used for energy basically. They will go through beta oxidation processes to yield acetyl coenzyme A, which can then go through the Krebs cycle, generate reduced NAD. The reduced NAD can then drive the electron transport chain, which will build up that proton gradient across the inner membrane of the mitochondrion. ADP synthase can then allow protons to move back across into the matrix, and will harness the energy released by the protons when they move down their chemiosmotic gradient, and it will use that to bind ADP to inorganic phosphate to create ATP. Okay, right, so that's how we have now delivered our long-chain carboxylic acids to the skeletal and cardiac muscle tissues during the fasting state. Okay, but the story doesn't end there, because what's going to happen to our very low density lipoprotein? Okay, so remember, our very low density lipoprotein started off as a very, very large molecule. Okay, so it was a big lipoprotein. It was between 28 and 70 nanometers. Okay, and it was bursting full of triacylglycerol molecules. Okay, well, what's going to happen is we're gradually going to lose these triacylglycerol molecules. That means that the whole thing is going to shrink. Okay, so you're going to have to get rid of some of the lecithin molecules or the other phospholipids that are within the phospholipid monolayer as well. Okay, so those will gradually leave as well. But what will happen is gradually the very low density lipoprotein will shrink. Now, it is on its way to becoming a low-density lipoprotein. It goes through an intermediate step, basically. It goes through a stage known as the intermediate density lipoprotein, or IDL. So this is called intermediate density lipoprotein, and this is sort of where the very low density lipoprotein has given away a huge amount of its triacylglycerol, but it hasn't quite given away all that it can give. Okay, so it's the sort of intermediate step basically. So it can still function, it can still go to the endothelial cells of capillaries within skeletal and cardiac muscle tissue, and it can deliver triacylglycerols to them. Okay, it still has some triacylglycerols, but it's smaller and it's on its way to becoming a low-density lipoprotein. Okay, so the final thing that you end up with once you have given away all of your triacylglycerol molecules is then a low-density lipoprotein, or an LDL molecule. So this stands for low-density lipoprotein. So basically, low-density lipoproteins are the very low-density lipoproteins once they have finished all of their useful role, basically. Okay, so sometimes they will be called the remnants of VLDL molecules. So they can be called VLDL remnants. Okay, so if we draw a picture of them, they are much, much smaller than the VLDL molecules. Okay, so they are between 20 and 25 nanometers in total, so they're smaller in size than the VLDL molecules. And also, nanometers, there we go. Also, they only have a single apolipoprotein. So basically, as the lipoprotein has been shrinking, we've acknowledged that lecithin molecules will gradually be leaving the lipoprotein. Okay, you will also have to give up some of your apolipoproteins, so they will also fall out and go into the blood. Okay, till the point where you have this LDL molecule, and the LDL molecule now only has a single apolipoprotein within its phospholipid monolayer. Okay, but this is a giant of a apolipoprotein. This is a big, big protein. Okay, this is almost 5,000 amino acids long. It's not quite there, but it's almost there. Okay, and this is apolipoprotein B100. So remember, this was one of those apolipoproteins that was initially in the VLDL molecule, but because the VLDL molecule was so much bigger, it didn't, dwarf the molecule in the way that apolipoprotein B100 dwarfs the LDL. Okay, it's the only apolipoprotein that the LDL molecule has room for, basically. Okay, so it's a big, big giant of the apolipoproteins. So LDL molecules have a single apolipoprotein within their phospholipid monolayer, and this is the apolipoprotein B100, and they have one of those. Okay, they then have a phospholipid monolayer, so here are some of these phospholipids in the phospholipid monolayer. And now what do they have within their lipid core? Okay, well, they've lost all the triacylglycerol molecules, so their entire lipid core consists of cholesterol esters. Okay, in addition, they also have cholesterol molecules within their lipid bilayer, which are free cholesterol molecules. Okay, for this reason, because these are effectively storages of cholesterol and cholesterol esters, people will often call LDL cholesterol. Okay? Right. So, we've now seen where the LDL molecules come from. We've seen that they are this product of the very low density lipoproteins, which are involved in this endogenous pathway by which we deliver lipid molecules to the skeletal and cardiac muscle tissues in the fasted state. Okay, what we now want to look at is, what do you do with these molecules now? How are they retaken up into cells, and which cells are they retaken up into? And for this, we're going to discuss the LDL receptor. But we'll start that discussion in the next video.